today we're going to be talking about epistasis. Epistasis is the expression of alleles at one locus that masks or modifies the expression of alleles at another locus. You can see this illustrated in the picture here, how if at locus 1, A will have a dominance over any other allele, so it will mask any locus at locus 2. If it is A recessive, it will not mask locus 2 alleles. Single dominant epistasis is when a dominant allele at one locus masks or modifies the expression of alleles at a second locus. So, if A is epistatic to big B and little b, then big A results in a particular phenotype no matter what is at the big B or little b locus. And this is when it can either be two big A's at the first locus or a big A, little a. If there's a, it will be recessive and the big B locus will still be present in the phenotype, but if not, the big A will have dominance over the big B allele. Let's consider a certain breed of sheep for the single dominant epistasis example. So in this breed, the white wool sheep is considered the normal offspring or dominant as we refer in genetics. And if the big A is not present at the first locus, it will result in the black wool sheep color because the black wool sheep color is recessive to white and it produces enzymes at locus 1 and 2. However, there's a third offspring probability, which would be the gray wool sheep. This is when both A and B are recessive. It produces that enzyme 1 and it would result in the gray sheep color. Here we have the phenotypes listed. You could either have big A, big B, or big A, little b, and still get that white sheep color. Or we have little a, big B for a black sheep, and little a, little b for the gray sheep. So let's consider a dihybrid cross example of two white heterozygous sheep, which would be considered our normal individuals for this population. So here we have their genotypes, and I went ahead and did two Punnett squares for each parent, and then we went ahead and placed the results from the Punnett square into a bigger chart, and we're going to go ahead and calculate like so. For example, let's just look at the first square. We have big A, big B, big A, big B, so that would result in two big A's and two big B's. And if we look back at our previous page, we would know that that would be a white sheep offspring. So after calculating for every square, you can see that we will get 12 white sheep and 4 of the big B dominant alleles. So that results in 3 black sheep and 1 gray sheep, as you see here, because there are 2 little b's and it doesn't have that big B there. So that would result in our 1 gray sheep, giving us the ratio of a 12:3:1.